Assalamualaikum. Today we will be presenting on unique yet highly dangerous bacterium called Clostridium botulinum. In terms of taxonomy, this species has kingdom of Eubacteria, phylum Bacillota, class Clostridia, order of Clostridiados, family Clostridiaceae, genus of Clostridium, species of Clostridium botulinum. Now let's talk about where this species can actually be found. First, it can be found in soil, especially agricultural or cultivated soil. Secondly, it thrives in lake, river, and marine sediment. Third, it can be found lingering in dust and decaying organic matter. And fourth, it resides within animal intestine and feces.
So next, I will explain the morphology of Clostridium botulinum. The spectrum is the gram-positive microorganism with a root-like shape. One important characteristic in that it is motile, meaning it can move using tiny structure called petrichous flagella that are distributed around the cell surface. So another notable feature is its ability to produce endospores. These endospores are highly resistant structures that help the bacterium survive extreme environmental conditions such as heat, dryness, and lack of nutrients. Because of these adaptations, Clostridium botulinum can remain dormant for long periods until conditions become favorable again. In addition, this bacterium is an obligate anaerobe, which means it can only grow in environments where oxygen is absent or very limited. These characteristics explain why it is often associated with improperly canned or vacuum-packed food. It is also commonly found in natural environments such as soil, sediments, and aquatic habitats. So now that we have discussed its physical characteristics, let's move on how Clostridium botulinum obtains in nutrients and energy. So, Clostridium botulinum is classified as a chemoheterotrophic bacterium. This means that it obtains both its energy and carbon from organic compounds found in its surrounding. Unlike organisms that use oxygen for respiration, Clostridium botulinum generates energy through anaerobic fermentation. This process allows it to survive and grow in oxygen-free environments. The bacterium also acts as a saprophyte, meaning it feeds on dead and decaying organic matter. By breaking down these materials, it contributes to nutrient recycling in the environment. However, under suitable anaerobic conditions, especially in improperly preserved foods, the bacterium can multiply and produce botulinum neurotoxin. This toxin is one of the most powerful biological toxins known and is responsible for the disease called botulism.
Now, I will discuss the general characteristic of Clostridium botulinum. Firstly, Clostridium botulinum is a gram-positive bacterium. This means that it has a thick bacterioglycan layer in its cell walls, allowing it to retain the crystal violet stain during gram staining. Secondly, it is a spore-forming bacterium. Under unfavorable environmental conditions, it can produce highly resistant endospores that enable it to survive extreme temperature, dryness, and nutrition depletion for long periods. Another characteristic is that it is motile. The bacterium can use flagella, which helps it to navigate through its environment in search for suitable growth conditions. In addition, Clostridium botulinum is an obligated anaerobic bacterium. It cannot grow in the presence of oxygen and thrives only in oxygen-free environments, such as sealed container or improperly canned foods. Lastly, it exhibits heterotrophic nutrition. This means it obtains energy and nutrition by breaking down organic matter from its surroundings rather than producing its own food. This characteristic allows Clostridium botulinum to survive in diverse environments and contribute to its ability to cause disease. Next, I will explain the unique characteristic of Clostridium botulinum. One of its most distinctive features in its ability is to produce botulinum neurotoxin. This toxin is considered one of the most powerful biology toxins known and specifically targets the nervous system. The production of this toxin can lead to botulism, a rare but potentially life-threatening disease characterized by muscle weakness, paralysis, and in severe cases, respiratory failure. Another unique characteristic is its association with improperly preserved food. The bacterium can grow in anaerobic conditions found in poorly processed canned food, fermented products, and vacuum-packed food where it produces the toxin. Interestingly, despite its toxicity, botulinum toxin has valuable medical application. In controlled dose, it is used to treat conditions such as muscle spasms, chronic migraines, excessive sweating, and certain eye disorders. It is also widely used in cosmetic treatments to reduce facial wrinkles. Furthermore, Clostridium botulinum is widely distributed in nature. Its spores can commonly be found in soil, dust, river sediment, and marine environment, allowing the bacterium to persist in many ecosystems. Overall, the combination of toxin production, spore formation, and environmental adaptability make Clostridium botulinum one of the most significant bacteria in both medical microbiology and food safety.
Hi, my name is Nariman Akila, and today I will be explaining briefly about the life cycle and reproduction of Clostridium botulinum. What is Clostridium botulinum? Clostridium botulinum is a bacterium commonly found in soil and aquatic environments. It is well known for producing botulinum toxin, one of the most potent toxin ever known. Clostridium botulinum reproduces asexually through a process called binary fission. During binary fission, a single cell first copies its own DNA, and then the cell then elongates and divides into two identical daughter cells. This is the main method by which the bacterium increases its own population. The life cycle begins with a vegetative cell, which is the active and growing form of the bacterium. When environmental conditions are favorable, such as when nutrients are available and oxygen levels are low, the vegetative cell continues to grow and reproduce continuously through binary fission. However, when the conditions become unfavorable, such as lack of nutrient, extreme temperatures, or too much exposure to the oxygen, the bacterium forms an endospore. An endospore is a highly resistant dormant structure that allows the bacterium to survive in harsh conditions for a long period of time. And then, when the environment becomes favorable again, the endospore undergoes germination and develops back into an active vegetative cell. The bacterium can then continue growing and reproduce through binary fission. So, in conclusion, Bacterium Botulinum reproduces only asexually by binary fission. Although it forms endospore, spore formation is a survival mechanism rather than the process of reproduction itself. This ability is to alternate between vegetative cells and dormant spores to help the bacteria survive in changing environment. That's all from me. Thank you.
Now let's move on to the importance of Clostridium botulinum to ecosystem. First, Clostridium botulinum helps in the decomposition of organic matter. It breaks down dead plants and animals in soil and aquatic environments, especially in places with very little oxygen. This decomposition process is important because it prevents the accumulation of waste materials in nature. Second, this bacterium contributes to nutrient recycling. During decomposition, nutrients such as carbon and nitrogen are released back into the environment. These nutrients can then be reused by plants and other organisms, helping to maintain soil fertility and ecosystem stability. Lastly, it is a part of natural microbial community found in soil and water ecosystem. Its presence helps maintaining the balance of microorganisms, which is important for sustaining biodiversity and healthy ecosystem functions. Other than that, one important economic contribution of Clostridium botulinum is in medicine and cosmetics. The botulinum toxin it produces is used in controlled doses to treat muscle disorders, migraines, and cosmetic procedures such as Botox. This has created a large medical and cosmetic industry worldwide. Next, Clostridium botulinum is important in the food industry because it is associated with food poisoning known as botulism. Food manufacturers must apply proper sterilization and preservation methods to prevent contamination, making food safety monitoring very important economically. Lastly, this bacterium is widely studied in microbiology and biotechnology research. Scientists use it to study bacterial toxins, the function, and disease prevention. Research on Clostridium botulinum has contributed to advancement in medicine and biological sciences.
Hi, my name is Nur Nawara Wadiha, and my part is Interesting Facts about Clostridium botulinum. Today we are going to look at its transportation as a myotoxic producer and explore its fascinating ecological role as a master of survival and environmental warning sign. To understand why Clostridium botulinum is such a formidable organism, we have to look at its primary evolutionary adaptation, the ability to form highly resistant endospores. When environmental conditions deteriorate, the bacterium shifts into a dormant spore state. Are practically microscopic fortresses. Can survive for years, even decades. Incredibly harsh environments such as dry soil and oxygen poor. This unique adaptation allows the species to persist across incredibly diverse ecosystems, safely waiting for the right moment to wake up. For example, scientists were recovering artifacts from the SS Central America, historic steamship that tragically dipped into the Atlantic Ocean. Presently, researchers recovered viable spores from the ocean sediment and items on board. Think about the conditions down there. The spores managed to survive for over a century, more than 100 years under crushing oceanic hydrostatic pressure, near freezing temperatures, and total darkness. They simply sat dormant, waiting for the right conditions to wake up. This case documented in the US Department of Agriculture in 2024 perfectly highlights why this organism is the ultimate survivalist. Because these spores are so widespread and sensitive to environmental change, Clostridium botulinum actually serves as a powerful indicator for climate change. Let's break down the ecological chain reasoning. First, we have the global warming effect. As global temperatures continue to rise, we are seeing severe droughts that dry out crucial wetlands. This leaves behind very shallow, stagnant, and oxygen-starved water. This brings us to a phenomenon known as a mass awakening. This specific hypothesis, warm conditions act as an environmental trigger, signaling to the dormant spores that it is the time to germinate. What follows is a massive spore germination cycle right in the heart of the nature. As a result, spikes in wildlife botulisms function as a stark ecosystem warning. When we see sudden outbursts of botulism in wildlife, it isn't just isolated disease. It serves as a critical biological warning sign of rapidly degrading ecosystem health. To ground this theory in real-world data, let's look at the case study of the Great Salt and wetland ecosystem. The process begins with a major climate shift. Rising global temperatures have driven historic droughts, causing extreme water evaporation in these vital habitats. As the water levels drastically drop, we get the physical blueprint for germination. The remaining shallow pools become incredibly warm, frequently exceeding to 220 degrees Celsius. Because the water is stagnant and warm, it rapidly loses its dissolved oxygen, becoming highly anoxic or oxygen deprived. This is the absolute ideal environment for an obligate anaerobe like Clostridium. Finally, we hit the bioindicator spike. These precise environmental changes immediately wake up the dormant Clostridium botulinum spores resting in the mud. Once awake, the bacteria multiply and produce deadly neurotoxins, triggering massive avian botulism epidemics. Historically, these seasonal events have been devastating, wiping out over 500,000 of waterbirds in a single seasonal outbreak. Ultimately, Clostridium botulinum shows us how microscopic survival mechanisms can scale up to cause massive ecological impacts under a changing climate change.